Chetna madam, hello.
Yes, Viju.
There is some echo. Someone is there who has again that entered in Zoom and
Hmm. Now also?
still echo is there.
It's coming. Hello?
Yeah, now it is clear and please,
Hello?
please on your video also. We'll see.
Hello, Viju.
Yeah, video on ಮಾಡು ಚೇತನ.
Speaker in the Hello?
Now the voice is clear. The video is not properly. Once again, ಅದು ಸ್ವಲ್ಪ ಇದನ್ನ camera fit ಮಾಡ್ರಿ camera.
Speaker
Camera adjust.
ಏನು ಹೇಳ್ತಾರೆ ಇಲ್ಲಿ
ಅಲ್ಲೆಲ್ಲ ಕರೆಕ್ಟ್ ಇದೆ ಸ್ಪೀಕರ್.
ಹೆಂಗ್ ಹೇಳ್ತಾರ ಎಷ್ಟೊಂದು ಫೋನ್ ಮಾಡ್ತೇನೆ
I can hear your voice clearly, but the camera ಸರಿ ಮಾಡ್ರಿ.
ಅದೇನ್ ಬೇಡ ಸ್ಪೀಕರ್ ಬೇಡ ಆ ಸ್ಪೀಕರ್ ಅಲ್ಲ ನಾವು ಕೇಳತ್ತಿದ್ದೆ
ಇಲ್ಲ, speaker is good. That voice is clear.
ವಿಜು ಕೇಳಿ
Camera adjustment. Camera adjustment.
ಹಾ ವಿಜು ಕೇಳಿಸ್ತೈತೆ ಆಡಿಯೋ
Now camera is adjusted. Audio is also clear.
ಹೊರಗೆ ಹೋಗಿ ಮಾತ್ರ ನಮ್ ವಿಡಿಯೋ ನೋಡಕ್
Ah, hello, Dr. Chetna. Can you hear me? Hello. Hello, Chetna madam. Can you hear me? Hello, Chetna madam. Can you hear me?
ಹಲೋ
Hello, Chetna madam. Can you hear me? Hello. Hello, Chetna madam. Hello, Chetna madam. Can you hear me?
ಬಂತು ಕಾವ್ಯ ಕಾಣತೈತಿ ಕರೆಕ್ಟ್ ಐತಿ ನಾನು ಮಾತಾಡ ಹಲೋ ಹಾ ಓಕೆ ಕಾವ್ಯ ಬನ್ನಿ ಇಲ್ಲ ಇಲ್ಲ ಹ್ಮ್ 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 ಹ್ಮ್
Hello. Hello, can you hear me?
ನನಗೆ ಕೇಳ್ತಿಲ್ಲ
Madam, can you hear me? Now you are doing it. ಅದನ್ನೇ ಸ್ವಲ್ಪ ಮಾಡಿ hello.
Hello.
हाँ मैम
Hello. I think that our voice is not audible to the speakers, so that's why they are trying it.
हाँ मैम
Once again, I'll check it. Okay. Now
श्योर मैम
they have started screen sharing. Dr.
मैम
Chetna madam, Dr. Chetna madam, can you hear me?
Ma'am, your voice is not audible in the PC.
Hello.
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 Yes, ma'am.
Yes, ma'am, it's audible.
Hello, Dr. Chetna madam, can you hear me?
Ma'am, they're unmute. Tell to mute, ma'am. Tell to unmute, ma'am. They're muted.
Yeah.
Hello. Now you can hear Dr. Chetna madam.
Hello. Bluetooth on ಮಾಡ್ಬೇಕಾ? ಬಿಟ್ಟು ಸ್ಪೀಕರ್ ಬಟನ್ ಎಲ್ಲ ಕವರ್ ಎಲ್ಲ ಇವ್ರು ಇಷ್ಟೇ ಇವ್ರು ನಾನು ಬರ್ತಿತ್ತು ಇವ್ರು ನಮ್ಮ ಕಾಲೇಜ್ ಒಬ್ರು ರೆಸ್ಪಾನ್ಸಿಬಿಲಿಟಿ ತಗೊಂಡು ಅಷ್ಟು ಜನ ನಿಂತು ಶುಕ್ರ್ ಹೋಗ್ಯಾನ ಹಲೋ
Yes,
ಹಲೋ
ma'am. Yes, ma'am.
ಕ್ಯಾನ್ ಯು ಹಿಯರ್ ಮೀ
Yes, ma'am.
Yes,
You
ma'am.
can hear that.
Hello, can you hear me?
Yes, ma'am.
Yes, ma'am.
Okay, then shall we start?
Hello, Dr. Chetna madam, can you hear me?
Yeah.
Yes. Okay.
Vijayalakshmi ma'am, can we start?
Yeah, doctor. Please start.
Yes, ma'am. Okay. Good evening. Good evening to one and all. Naneshwari, over to you.
Good evening to all. Jignasa AIAPGET orientation class, Jai Shri Gurudeva. Success in AIAPGET is not determined by intelligence alone. It is built on the foundation of clear guidance, consistent preparation, and unwavering determination. Every successful postgraduate journey begins with a single step in the right direction. The AIAPGET orientation class, Homeopathy, is a dedicated, innovative design to help aspiring postgraduate students understand the examination, develop an effective preparation strategy, and approach their goal with confidence. This session aims to provide valuable insight into the AAPGT examination pattern, subject wise planning, time management, preparation techniques, and the mindset required to excel. Led by the experienced mentors and academicians, the orientation will equip participants with practical strategies to maximize their preparation, avoid common pitfalls, and stay motivated throughout their journey. Whether you are just beginning your preparation or looking to refine your approach, this orientation will serve as the ideal starting point to transform your aspiration into achievements. Jo- Join us as we take the first step towards excellence in AIAPGET and embark on a journey of learning, perseverance, and success. Jignasa is a pan Indian movement dedicated to creating a unified platform for addressing all dimensions of the Ayush system: Ayurveda, Yoga, Unani, Siddha, Sovarikpa, and Homeopathy. The motto is Learn Ayush to Practice Ayush. Jignasa Karnataka starts as a pioneering organization among Ayush students in the state. Known for its innovative ideas and impactful programs, we have successfully organized landmark events such as International Conference and National Arogya Fair in 2018 and 2022, and Yantra in 2023, and Mantana in 2024 in collaboration with NIMAS. For the homeopathic fraternity, Unnati 2024 was a unique platform dedicated to empowering homeopathy students. We have actively partnered with the government of Karnataka in Yoga Day celebration, and our flagship event, Achintya Ayurveda, has emerged as a leading conference in the field of Ayurveda across the state. To promote traditional cow based health practices, we have consistently conducted Panchagavya workshops, training students and practitioners in the preparation and benefits of cow derived products. Reaching out to underserved and rural areas has been a priority. We have conducted numerous health camps, awareness programs, and Swarna Bindu Prashana drives aimed at improving children's immunity. In addition to support Ayush students during their academic journey, Jignasa Karnataka has continuously organized online classes, exam preparatory sessions, and career and postgraduate guidance programs, fostering clarity and confidence among the graduates aspiring for the higher education and professional excellence in the Ayush domain. Dr. B.D. Jati Homeopathic Medical College, Hospital and Postgraduate Research Center, Dharwad, was established in 1992 to provide quality homeopathic education and healthcare. Located on a 5-acre campus in the heart of Dharwad, just 1 kilometer from the central bus stand and railway station, the institution offers modern infrastructure, well-equipped laboratories, a rich library, and experienced faculty under the leadership of Principal Dr. Anand J. Hosur, sir. The college is permitted by the Ministry of Ayush, Government of India, Government of Karnataka, MARBH, and the National Commission for Homeopathy, and is affiliated with Rajiv Gandhi University of Health Sciences, Bengaluru. The institution has been awarded an A+ grade in the QCA-NCH ranking for its excellence in homeopathic education and quality healthcare. Every successful journey begins with the guidance of someone who has walked the path before us. Today, we are privileged to be in the presence of an inspiring mentor whose knowledge, experience, and dedication have guided countless students toward academic excellence. It is an honor to welcome a speaker who is here to inspire, enlighten, and empower us on our AAPGT journey. With great respect and admiration, let me introduce you all to our esteemed speakers. It is a privilege to introduce Dr. Chetana Sivai ma'am, MD Homeopathy, an eminent academician and dedicated educator in the field of homeopathic medical education. Dr. Chetana ma'am currently serves as professor and head of department Human Physiology and Biochemistry at Dr. B.D. Jati Homeopathic Medical College and Hospital, Dharwad, Karnataka. With an impressive 27 years teaching experience, she has played a pivotal role in shaping generation of homeopathic physicians through her commitment to academic excellence and student mentorship. She has further enriched her expertise with a diploma in yoga and diploma in food and nutrition, reflecting her holistic approach to health and wellness. As a recognized research guide, she has actively contributed to the advancement of homeopathic research and has published scholarly articles in the National Journal of Homeopathy. She also serves as member of editorial board of her institution's in-house magazine, contributing to academic and literary development. In recognition of her exceptional dedication to teaching and academic service, she was honored with the Best Teacher Award in the year 2025. Today, we are honored to have such an accomplished educator and mentor with us. On behalf of everyone present, I extend a warm and heartfelt welcome to Dr. Chetana Sevai ma'am. We are eagerly looking forward to an enlightening and inspiring session. Thank you for joining us, ma'am. It is my distinct privilege to introduce our one more guest of honor, Dr. Manjula S. Haleholi ma'am, an eminent homeopathic academician, clinician, researcher, and educator. Dr. Manjula S. ma'am is professor and head of Department of Homeopathic Pharmacy at Dr. B.D. Jati Homeopathic Medical College, Hospital, Post-Graduation Research Center, Dharwad. She holds an MD in Organon of Medicine and Homeopathic Philosophy and is currently pursuing her PhD in homeopathy. With 23 years of teaching experience and 27 years of clinical practice, she has made significant contribution to homeopathic education and patient care. She has successfully completed advanced faculty development programs in competency-based medical education, curriculum implementation, pharmacovigilance, and education methodology. A dedicated researcher and mentor, has guided undergraduate research projects, secured research grants, presented papers at national and international conferences, published articles in peer-reviewed journals, and serves on the educational board on her institution in-home magazine. Her academic excellence has further been recognized through appointments as a resource person for BHMS curriculum redesign, faculty member of Rajiv Gandhi University of Health Science, question paper setter, examiner, and inspector for homeopathic institutions. Today, we are truly honored to have such an accomplished academician and inspiring teacher with us. On behalf of everyone present, I extend a warm welcome to Dr. Manjula ma'am and invite her to deliver her valuable session. It is my great pleasure to introduce one more guest of honor, Dr. Sushmita KB, MD Homeopathic Pharmacy, who serves as an assistant professor in the Department of Homeopathic Pharmacy at Dr. B.D. Jatiya Homeopathic Medical College, Hospital and Research Center, Dharwad. Dr. Sushmita ma'am is a dedicated homeopathic academician with a strong foundation in homeopathic pharmacy. She is continued to excel in teaching, academic development, and nurturing future homeopathic physicians through evidence based learning and professional guidance. It is our privilege to have her with us today. On behalf of everyone present, I extend a warm welcome to Dr. Sushmita KB ma'am and invite her to deliver her valuable session. Thank you all. Over to Chetana ma'am.
Thank you, Dhaneshwari. Okay, shall we start?
Yes, ma'am.
So
Yes, ma'am.
thank you, Jignasa team. Thank you, Anand sir. Thank you, my colleagues and my BD Jati team for giving me this platform. Myself, Dr. Chetana, Department of Physiology, is over to you right now. So everybody knows about the pattern and regarding AIAPGT. So we are not repeating it. It may be boring for you, but still we'll go with it. So mode of exam, you all know it's an online pattern. Language is English. And total questions, you'll be having 120 questions. So out of this, 60 will be for the core subjects and 60 will come for the allied subjects. Right. And correct answer will give you +4 and wrong answer -1 and unattempted 0. So as I said, subject wise, it is 60 for core subjects and 60 for allied subjects. So I won't use the word as allied subject because it is a parental subject. First year subjects, whether it is anatomy, physiology, pharmacy, they are all foundations. So based on this foundation only, the all other second year, third year, final year subjects will be carried on. And let's not use the word as allied subject. It's a parental subject which is having an orientation with all other subjects. So we orient with pathology, medicine, and especially materia medica, repertory. And the word what we are using here is priority. Let's not use it as a priority over here because we are not having very high, medium, moderate, lower priorities. All questions are priority questions only because when you are attempting AIAPGT examination of 120 questions, you are not solving the questions as very high, medium, moderate, lower. You are solving the questions. That's it. Whether it is anatomy question, physiology question, pharmacy question. So each question holds you 4 marks. So let's not use the word as priority. All 120 questions hold the high priority for you. And based on our 2024, 22, 23, 25 questions, so we have found out some focus areas where mostly we are getting questions on general physiology. High priority is given for cardiovascular system, nerve muscle physiology and blood, where around 15% of the questions will be getting from those and endocrine. And less priority, what we are seeing in the question papers is the general physiology and renal physiology. So as you all know, as I already said, the foundation is always anatomy and physiology. From this only, we have all the pillars of second year, third year and final year and integrated subjects, all integrated subjects like patho, medicine, repertory, materia medica, organon. So based on this only, if you are strong in your foundation, then only you can solve all the questions, right? And AIAPGT weightage for our physiology and biochemistry because our physiology never goes alone. It moves with biochemistry also. If we are having like 5 means exactly 5 questions, we don't have any pattern like we are marked with 5 questions for physiology and anatomy. It is approximately plus or minus. We may get 7 questions also, we may get 4 questions also. So it's not clear about, but based on all previous question papers we have seen in physiology, 3 to 4 questions, and in biochemistry 1 to 2 questions, definitely we used to get it. And frequently tested physiology topics, as you all know, are cell physiology, blood, nerve muscle physiology, CVS, respiratory, renal, GIT, endocrinology, reproductive, special sensors, temperature regulation, and exercise physiology. And on biochemistry, major what we could see in previous question papers is usually enzyme based. And vitamins, carbohydrates, protein, fat metabolisms, molecular biology, acid-base balance, all based on all these we have got the questions. So high yield areas for both physiology and biochemistry based on previous question papers. Maximum questions we used to get from cardiac output, stroke volume. It's not compulsory that we'll be getting from these chapters only, but since now we have been focusing the questions from this only, and oxygen transport, CO2 transport, dissociation curves, acid-base disorders, GFR and renal clearance, hormones and the feedbacks, nerve conduction, muscle contraction, and this is very important regarding respiratory. From first year you have studied volumes and capacities, right? And blood groups and their coagulation, enzyme kinetics. Usually, you won't give much importance for biochemistry. I know that, but we are getting questions from enzyme kinetics. And we are getting questions from TCA cycles, oxidative phosphorylation, liver function, bilirubin metabolism. Especially, we are getting questions based for conjugated and unconjugated. We'll have very small discussion on those things in further things. So basically, we can't go on with all the syllabus of physiology. Let me tell you, you people, when you are preparing for AIAPGET, prepare some flashcards for your quick study. Here you can't hold the volumes of books to prepare for the examination. So keep some flashcards, especially for physiology. I recommend you for all the normal physiology values. Why? Because you will be getting these questions for your MCQs also, for match the following also, and even for your statement verifications, AR questions you will be getting. We have seen it. So I'm telling you, so prepare some flashcards where you prepare it in right hand so that in the last moment you can have a look on it. I know you will be concentrating on establish the major like mathematical, organon, respiratory, medicine, right? But don't forget the base or the foundation, because if you understand concepts over here, because the physiology is not a memorization, it is conceptual study. You have to understand the concept, then only you will be able to move for your medicine, pathology, respiratory, mathematical, all OBG surgery. So I recommend you go for normal physiology value flashcard, some of the normal values from cardiac system, respiratory system, chapter blood and renal function. You can have a gross look on it, like cardiac output, heart rates, blood pressure, stroke volumes, ejection fraction. This has been asked many times. Ejection fraction, respiratory, as I said, total volumes, like lung volumes and lung capacities. You prepare in a simple way. You prepare it, so it will be helpful for you. And blood का तो तुम लोगों को पता ही है. And in renal physiology, ECF. So ECF means external fluid. So where total body fluid will be 60%. Out of that, 20% will be external and 40% will be internal. So you should know all these things for MCQs. Even for match the following, they have asked all these questions, right? And next, I want you to go for some of the formulas, because they have been asking you formulas in previous question papers. So you concentrate. That's what I'm telling you. You cannot read every chapter. So prepare some flashcards, fast, quick, and you have to have a look before examinations. So we have a few formulas, important formulas like minute ventilation, alveolar ventilation, vital capacities, inspiratory capacity, functional, total lung capacity, oxygen content, oxygen capacities, percentage saturation. So all these things, you should know the normal value, plus you should know the formula. Because as I already told, it's a conceptual study. So only these things, if you understand and if you keep in your mind, then only you can move for others. This is a base. Right. Then in cardiovascular system, important formulas, as you all know, stroke volume, cardiac output. So it is a heart rate into stroke volume, ejection fraction, and pulse pressure. Pulse pressure is systolic blood pressure minus diastolic blood pressure and mean arterial pressure. So you have to, because there is no other way. You cannot skip this. This you have to and must read it. And total peripheral resistance. So all these things better I recommend you go for some charts, prepare handy charts so that it will be easy for you in the last moment. And why I'm recommend you for cardiovascular system is we have been asked questions based on this. So like I'll tell you, cardiac is directly proportional to heart rate and stroke value. So in statements like AR questions, they'll ask you. So they'll say cardiac output is directly proportional to heart rate. Then reason you have to give. So in reason, they may give it as it may be due to the left ventricular output, or they may give it as a right ventricular output. Whether it is right or wrong, you should know. It is in left ventricular output. It's the right answer. So for all those things, you should know whether it is directly proportional, whether it is inversely proportional. These are the few basics you are supposed to read out. And one more thing in one-line revision, this is like we have few cycles in our syllabus. So that is, you all know cardiac cycle, menstrual cycle, Krebs cycle, urea cycle, and Cori cycle. So in simple one-line memory, you have to keep this in mind. What is cardiac cycle? Because this has been asked for your match the following. So filling, contraction, ejection, and relaxation. Again, filling, contraction, ejection, and relaxation. So cycle means the start point and the end point will join the start point again. Likewise, in menstrual cycle, we have the phases of menstruation, follicular phase, ovulation phase, and luteal phase. Come back again for the first, that is menstruation. So like this, for all this, you have to have a memory, quick memory. And Krebs cycle, I think you have studied in flowchart mechanism. That is the citrate, getting converted into isocitrate, then alpha ketoglutarate into succinyl coenzyme, then succinate, fumarate, malate getting converted into oxaloacetate. So this oxaloacetate again comes into the starting point, where again you will be having the same cycle, repetition of the cycle. Urea cycle starts with ornithine getting converted into citrulline, arginosuccinate, arginine, and the end product will be urea plus ornithine, where again that ornithine will be utilized in the cycle. Likewise, Cori cycle will come for your muscle physiology, where again in muscle we'll be having the deposition of this glucose as a lactate, which gets converted into again glucose, glycogen. So this cycle from muscle, liver, so this continues. So these cardiac cycles, menstrual cycle, Krebs, urea cycle, Cori cycle, these few cycles you keep in mind. Now coming to, so you have few graphs in our physiology. You all know about these graphs. Why we have to study these graphs is, these are important because either they'll ask you for the identification of the graphs, or they will ask you for some MCQ questions, AIAPGET questions. So we have graphs in, so we have biggest graph also called as cardiac cycle. So what you can weave in this graph is the pressure changes and the volume changes, right? And in oxygen hemoglobin dissociation curve, you will be having a sigmoid shape curve, and you have an action potential curve where you will be having depolarization and repolarization. And we already discussed regarding enzyme kinetics. You will be coming across two important graphs, that is Michaelis-Menten curve and Lane-Weaver plot. Why I am concentrating on these two curves are, this Michaelis-Menten curve and Lane-Weaver plot, both are related for the enzyme activity. Michaelis-Menten curve, just remember, so Michaelis-Menten curve is for enzyme activity, whereas Lane-Weaver plot is for enzyme inhibition. These are the questions which they will be asking you for the statements, AR statements. So you know that AR statements, that is assertion and reasoning. So just in a quick glance, I will tell you, like in Bigger's diagram or cardiac cycle diagram, you will be having pressure and volume changes graph. That is, graph will be having waves, like ECG waves. And oxygen hemoglobin, it's a sigmoid shaped, S-shaped curve. And action potential, again you have depolarization and repolarization curve. Michaelis-Menten curve is a hyperbola curve, and Line-Weaver-Burke plot curve is a linear curve. So if these curves are given for identification, so you should be able to identify these curves. They are the simple curves. So as I already said, and you should also know quick difference between oxygen dissociation curve and carbon dioxide dissociation curve. I know you know that, but still, the major differentiation over oxygen dissociation and carbon dioxide dissociation is the shape of the curve or shifting of the curve. In oxygen dissociation, the curve shifts to right side when there is increased pressure of carbon dioxide, temperature, and reduced pH. So here we'll be studying about the shifting of the curve to the right and left, right? So when you are studying about the carbon dioxide dissociation curve, you are studying about the curve moving to up and down. So curve shifting to upward and downward. This is the major differentiation. And how you really study these curves is, we have this oxygen dissociation curve like Bohr effect. We call it as based on Bohr effect, and carbon dioxide dissociation curve will be we call it as an Haldane effect. Just remember this, and the next few slides we'll discuss regarding this also. Already I told you in enzyme kinetics, major question what we have already come across are like Michaelis-Menten curve and Line-Weaver-Burke. Already this I told this is a hyperbola curve, and Line-Weaver-Burke plot is a straight line. It is and all graphs, whatever you are saying, just keep in one single word. This Michaelis-Menten is related for our enzyme activity, whereas Line-Weaver-Burke is for enzyme inhibition. Again, I stress you, such kind of questions you will be getting for your AR questions. That is assertion and reasoning questions. These are like, we may feel they are very simple questions, but when you are finding the answers for multiple choice answers for your AR clarification or explanation, so you have to be very careful and very quick in answering, because already you know you have 120 questions and 120 minutes. You should be very quick. You can't waste time over there. Intelligence is second thing. So how quick you are in answering is, that's all that matters over there in your examination. And we have another slide discussion which I feel very important in neuromuscular physiology is action potential, which you have come across in muscle physiology, in cardiovascular system, and in central nervous system. So here, four terminologies I'll give you a very quick glance. That is action potential starts after resting membrane potential. So all our muscles will be at the resting state. So this resting membrane potential will be taken over with a slight stimuli. When there is initiation of this stimuli followed by the response, we have action potential. This action potential where we'll be having contraction followed by depolarization, repolarization, and this action potential again goes for the refractory period. What is this refractory period? Refractory period is a state after action potential, and this will end with resting membrane potential. So after action potential, our muscles will be completely in contraction state. So that cannot stay forever. So, and before resting membrane potential, they come to refractory period. In refractory period, you have again two stages. That is not, I won't tell it as a stage, two patterns are there. One is absolute refractory, another one is relative refractory. Absolute refractory means what? after action potential, even if you give a stimulus, the second action potential will not be generated. That's for sure. Absolutely no second action potential. Whereas in relative refractory period, sometimes, in some cases, if second stimulus is higher than the first stimulus, then definitely second action potential is generated. But by rule, it's not a good thing. So these things we may see in heavy exercises. Right now, all are exposed for these heavy exercises, gym training. There we are finding all these things. Better keep our cycle only with resting membrane potential, action potential, and absolute refractive period. When you give more stimulus, then definitely it is a strain for our muscles, and we may in future face the problems. So I think you are quick in memorizing resting membrane potential, followed by action potential, followed by absolute refractory period, where no second action potential is generated, and relative refractory period, second action potential is generated, provided the stimuli is little higher than the first stimuli. So I already told you in the chapter liver regarding unconjugated and conjugated, and you will be getting a lot of questions in pathology also, in medicines also. So have a very brief introduction of this unconjugated and conjugated. After the breakdown of old RBCs, so the bilirubin what is formed is called unconjugated bilirubin. This is an indirect. This is unconjugated indirect bilirubin, what we are calling. So this is before it enters the liver. Means before it enters the liver, we call it as an unconjugated, and it will be traveling with one protein. No doubt it will be traveling with the protein, and it cannot be excreted in the urine, and it is water insoluble. This you keep in mind. So the unconjugated bilirubin, which we get after the breakdown of the RBC, is water insoluble, and it walks with the albumin to the liver. And this is not a processed bilirubin. This is unprocessed bilirubin. We call it as unprocessed. Once it enters the liver, it gets processed in the liver. It gets processed in the liver with an glucuronic acid, and then it becomes processed, called as conjugated bilirubin. So here you should keep two things very clear. Unconjugated bilirubin is an unprocessed, water insoluble, and which is albumin bound, which is there in the blood before entering liver. After entering liver, it gets processed as an conjugated, which we call it as a direct bilirubin, and it gets combined with glucuronic acid and becomes an water soluble, which is excreted in the bile. So it is excreted in the stools as stercobilinogen and excreted in the urine as urobilinogen. So I think you are clear about this unconjugated bilirubin and conjugated bilirubin. And let's conclude this slide with a word. Whenever there is excess RBC breakdown in the body, then we will be having excess of unconjugated bilirubin in the circulation. Whenever we have some liver diseases, then we will be having conjugated bilirubin in the bloodstream. So be clear, liver is affected, you will be getting conjugated or direct bilirubin in the bloodstream. And when there is excess of RBC breakdown, so we will be having an unconjugated in the bloodstream. So quick glance I want you to have on Bohr effect and Haldane effect. Again, this is a concern for your AR questions. So that is assertion and reasoning questions. I am telling you again and again, Bohr effect and Haldane effect. Very quick, how to differentiate over here is, in Bohr effect, you will be having hemoglobin where the oxygen leaves hemoglobin. Be clear, oxygen is going out of the hemoglobin, whereas in Haldane effect, what happens? Oxygen will bind to the hemoglobin so that carbon dioxide will be released. So be very clear, in Bohr effect, oxygen moves out from the hemoglobin, whereas in Haldane effect, oxygen gets bind to the hemoglobin so that CO2 will be released from the lungs. I think you are clear in this. So types of questions you all know: single best answer, MCQs, match the following, two statements, that is statement 1 and statement 2, and you will be asked for whether they are correct or not, and assertion AR questions. Like assertion question, you all know they will be asked in MCQs, where both assertion may also be correct and reason may also be correct. So it is the relation between two statements. Just one example I'll give you. So the question may be, insulin lowers blood glucose, and reason is it increases glucose uptake by skeletal muscle and adipose tissue. So the reason is right. The question they will ask you for MCQs is A and R both are correct. A is correct and R is not the reason given for A. Like that you will be getting the questions. So like SA node acts as pacemaker is one statement, and the reason is it has got the highest spontaneous depolarization rate, pacemaker. So the reasoning is correct. They may give you wrong reasoning also and ask you to give the correct answer. So quick glance for you people to see in last 2025 AIPGT questions which were covered were from cardiac physiology, endocrinology, renal physiology, and special senses and CNS. And in 2024, core topics tested were synaptic transmission mechanism, endocrine feedback, renal physiology, and nervous system and cell functions. And in 2023, they covered respiratory, measure, then cellular physiology. Little questions we got from renal also. Then 2022, they touched nerve muscle physiology, respiratory chapter, and a few biochemical markers. So if you all are alert, just go through the question and let's see. Okay, so try to answer. Try to answer. Yes. So answer is principal cells. The cells in the last part of distal convoluted tubule and in the collective dust that absorb sodium and are regulated by ADH and aldosterone are principal cells. So match the following. This is related for our cardiac system. This is from the question paper only. We have taken it out from the question paper. If possible, the aspirants, AIPGT aspirants, be quick and answer it. Be quick. So this has been asked for you. And the answer is second. And again, they have asked you that body mass index formula. So only I told you, you make some flashcards of formulas. So you will be able to have. So the right answer is fourth one. Body mass index will be having weight divided by height square. Austin Flint murmur is heard in. That is heard, it's heard in IoT regurgitation. Which of the following and why does cerebrospinal fluid contribute to homeostasis? They have asked you the ways. See, though you are taking physiology very easy, but they are making you to, yes, definitely you have to scratch your head for these things. See, when they are asking ways means all options you have to read and you have to arrange it. Match the following also you have to arrange, and this also, this is 2. This is mechanical protection followed by chemical protection followed by circulation nutrients. Though they are tricky, you people will be stuck with anatomy physiology only. So I wish all of you score more marks in those questions. The clinical condition, phecochromocytoma, results due to which hormone produced in excess? No doubt, even pathology, your medicine, everything carries these questions. That is noradrenaline and adrenaline, because it is related for our adrenal medulla. Which of the following statement is not true regarding sensory neural hearing loss? So answer 4. I'm a bit quick over here because I feel you have gone through these questions in your previous question papers, and a few more from 2024 I'll share. So enzyme secreted by the kidney that is involved in the control of blood pressure. Quick, your answer should be very quick. Answer 4, renin. I know everybody is very quick in your medicine and pathology. Ependymal cells are the cells that secrete cerebrospinal fluid, that secrete, maintain myelin in the peripheral nervous system, whose food processes from the blood-brain barrier, and that remove bacteria and damaged tissue in the. So see, what I want you people is, before answering, read the question properly. Ependymal cells are the cells. Answer 1, they secrete cerebrospinal fluid. So given below are 2 statements. One is labeled as assertion and another one is labeled as reason. So assertion, this is from the question paper I am telling you. It's not prepared by me. So focus on this. Water soluble hormones act on its target cell through second messenger system. Reason what they have given is the hormone receptor complex on the cell membrane activates G proteins, thereby activating adenylate cyclase and catalyzing conversion of ATP to cAMP, which activates protein kinase. So here what you should remember is water soluble hormones. Before entering the target cell, they have to cross the lipid layer. They cannot cross lipid layer alone. So they need a second messenger. So the reason what they have given is correct. So both statements A and R are correct. That's what the explanation is. Because the water soluble hormone has to cross the lipid cell membrane. It can't enter directly. It has to enter with one second messenger. So if you know the concept, physiology is conceptual based study. I am telling you again and again. Now, place the given stages of phagocytosis in the correct order. Be quick. Quick, you are given only 30 seconds. You know the process of phagocytosis? All of you know the process? So it is like you are having answer as second one. Why? So the steps are chemotactic attraction of phagocyte, followed by adherence to microbe, followed by ingestion to form phagosome, and followed by formation of phagolysosome, followed by destruction of the microbe. See, physiology you are thinking. हाँ, MCQ में 1-4 ऑप्शन रहता है, कौन सा भी हम लोग मार्क कर सकते हैं? No. The questions what we have been seeing from 24, 25, 23, 22 are all based on your understanding of the concept, and you have to be very quick in answering during your examination. See again, these are all questions from the question paper only. Which of the following are true? The sites of olfactory transduction are the olfactory hairs. The olfactory bulb transmit impulse to temple. To read only, this will take you 60 seconds. That's why I am telling you, you have to be very quick in reading your question and very appropriate, and then you have to arrange it also. The answer is the site of olfactory transductions are the olfactory hairs, then followed by the axon of olfactory receptor pass through the olfactory foramina in the cribriform plate, and then first order neuron followed by second order neuron. Complete concept if you know, then only you will be able to. So 60 seconds is not enough to answer your questions. So just conceptual based study, you concentrate, and some clinical scenario questions also you'll be getting, AR questions. So flashcards you prepare of normal values, graph interpretation, and prepare some flowcharts also. I want you to prepare some few quick flowcharts, right? And preparation strategy, very good books are there, Genong, Guyton, but Shambulingam you have studied and you will be stick to it. I don't say anything for you. And many other books are there for your MCQs. I recommend you, AK Jain is there. And you all already know that AIAPGET books, that is Success Guide on Homeopathy and Success Homeopathy. Again and again, I am pressurizing you. You concentrate on physiology and anatomy so that you will excel in your patho and medicine questions, right? And recommended books, many are there. You are also referring many, but you follow the previous year MCQ question papers and few of mock test. Mock test is very important for you. Attend as much as mock test. And we are also conducting mock test. Attend those mock tests. That is very must, because I know you cannot complete all the syllabus from first to final year so quick. I wish that you attend only MCQs. It is my personal view. And you know all the revision concepts, as I already told, concentrate on definitions, formulas, values, and flowcharts. Final advice I want to give you is concentrate on conceptual study, practice MCQs, daily you practice, revise repeatedly, memorize normal values, graph interpretation, solve previous AIAPGET papers, and attend mock test. Continuous mock test you have to. So key message for my students, so those who are aspiring to crack AIAPGET, master concept rather than memorizing isolated facts, so that this is the foundation on which it is based. So thank you one and all. I'm very happy for all the participants who have participated. Right, I think you all are benefited, and now I request Dr. Manjula Madam to take over the seat. Good evening, students. Okay, am I audible? Right. After brainstorming physiology, we have a short, quick, crisp homeopathic pharmacy. Welcome to the homeopathic pharmacy orientation. is fixed, that is on 22nd. Few days only left, right? So marking scheme and all, everything you know. Let us go to the next. So homeopathic pharmacy, what I call as, it is a chota pataka, bada dhamaka. Means here the syllabus is very less. Okay, but the scoring is most scoring subject. If you give your 2 to 3 days for this homeopathic pharmacy, definitely 12 questions means 48 marks are in your pocket, right? Sometime in the last previous year question papers, even up to 15 questions they have asked. So 60 marks are in your hand for revising homeopathic pharmacy for 2 to 3 days. And already you know the types of questions. Let us skip it. At the end of the session, you are able to focus areas, recommended topics, and solve the previous year question papers. So focus areas are basics of homeopathic pharmacy, pharmacopoeia, preparation of homeopathic drugs, sources of homeopathic drugs, vehicles, and potentization. Coming to other recommended topics: prescription, legislation, which includes Drugs and Cosmetic Act, PHYSIOLOGY, and previous year question papers are also solved here. Homeopathic pharmacy. Here few definitions, if you remember. Okay, they may be asked as a MCQs. So pharmacon means drug, and pharmacology is study of the drugs, which includes subject of pharmacy, therapeutics, and materiality. And coming to the pharmacognosy, gnosis means what? It is a knowledge. Pharmacognosy means the knowledge which helps for the identification of the drugs. And pharmacodynamics, which is the dynamic action of the drugs. Then continue with basic terminologies. Pharmaconomy. Nomy means the way, the management, or the principle. So the roots of administration of drugs is pharmaconomy. Then pharmaceutics, which means preparation of the medicine. Pharmacophilia. It is a feeling for to take the drugs. Some people will be having the affinity towards the drugs, but it is not like the mania. Whereas in pharmacomania, where there is an irresistible desire, whenever they see the medicine, they will take that one. That is what is a pharmacomania. Whereas pharmacophilia, if they have affinity, they will take the medicine with the love and affection. Okay. Then pharmacophobia, it is fearful to take the medicine, is pharmacophobia. And something different here, extemporaneous pharmacy. Extemporaneous pharmacy is according to the physician at the dispenser. For example, I want to give a sulfur 200, but I may ask the dispenser to do it either in the water dose, or in the Saclac powder, or in the globules form, or I may ask him to succus more than 10. It is according to the dispensing physician at his dispenser. So that is what is a extemporaneous pharmacy. There are so many other terminologies are there which are related to the pharmacon, that is with the drug. So please give importance to that one and study it. So in 2017 AIAPGET, they have asked, just go through the question and answer. Please answer it. Which deals with the dynamic action of the drugs.
4.
What is that? What is the answer? Anybody? So the answer is, what is the answer? Pharmacodynamics. So which is dealing with the dynamic action of the drugs on the human beings. Right. Now, pharmacopoeia. Pharmacopoeia, it is a small 2 to 3 pages of pharmacopoeia is being explained in your Mandal and Mandal pharmacy. Please go through it, because many permutation and combinations have been asked from the pharmacopoeia. It is a supreme authoritative book published by any government. It is an official pharmacopoeia. Unofficial pharmacopoeia means which is published by any person or by a company like Wilmar Schwabe. They have got their own pharmacopoeia, or any other person or NGOs, they may publish the pharmacopoeia, which also includes either in the official pharmacopoeia or in the unofficial pharmacopoeia. In both, it is starting from the identification, preservation, preparation, and standardization of the each drug, which is being called as monograph. Monograph means each single drug has been explained in a pharmacopoeia. The first pharmacopoeia was published by German Homeopathic Pharmacopoeia in 1825 by Carl W. Caspary. This has been asked in the question, right? Please note it down. Next, this official pharmacopoeia, I have arranged it in a chronological order. So this chronological order has also been asked in a match the following questions. Okay, these are initial first publications, and recent question paper also been asked with the recent editions. So please concentrate with the first publication as well as recent editions. So German Homeopathic Pharmacopoeia, British Homeopathic Pharmacopoeia, Homeopathic Pharmacopoeia of United States, French Homeopathic Pharmacopoeia, Homeopathic Pharmacopoeia of India in 1971, and unofficial pharmacopoeia. First unofficial pharmacopoeia in India was published by M. Bhattacharya and Company in Calcutta in 1893. And Encyclopedia of Homeopathic Pharmacopoeia by P. N. Verma and Hindu Ved is also available. And other countries' unofficial pharmacopoeia like Brazilian, Mexican, Pakistan, these have not been published by their government. That's why they are called as unofficial pharmacopoeia. So they may ask you in the question paper all these three and one more official pharmacopoeia, and they may ask you to identify which one is that. And I would like to ask you that by heart this table of Homeopathic Pharmacopoeia of India, HPI, which was begin with the Dr. B.K. Sarkar in 1971 with the 180 drugs. Why I am asking you? Many permutation and combinations have been asked in the from the question Homeopathic Pharmacopoeia of India. They may ask you when third volume published in which year, or how many monographs are there in the 8th volume, like that, and 11th volume is published or not. All these type of questions have been asked. So in 2017, direct question is there. First volume of Pharmacopoeia was published in which year? Will you answer? It is very direct question and easy question. Anybody? Yes. Yes, it is 1971. Very good. So, very good. Answer is 1971. Yes, here. Correct statement regarding Homeopathic Pharmacopoeia of India. First volume had a monograph of 180 drugs. If you remember, yes, it is correct answer. Third volume was published in 1988. Yes or no? No. It was published in, very good, 1978. First chairperson was J.N. Kanjilal. Is it Kanjilal was there? Please answer. It was B.K. Sarkar. Yes, B.K. Sarkar. So B and C are wrong. And HPI is included in second schedule of the Drugs and Cosmetic Act 1940. So which option is correct? Correct answer. Answer is option number 1, A and D only. Right? Now coming to the preparation of homeopathic drugs. So preparation of homeopathic drug, first one is in the old method or in the new method, these terminologies has been asked and these definitions are also been asked in the MCQs. Let us go through it. A crude mixture of drug substance thin pasty state before the preparation is called as magma. I would like to give an example. In preparation of mother tincture, when thin pasty state is formed, before the formation of mother tincture, that pulp is magma. Or simple layman language, before preparation of biryani, what you make that masala, no? Remember, that is what is the magma. Don't write biryani as a answer over there. It is magma. Next is mud. Inert fibrous insoluble material after the preparation. Means once the mother tincture is prepared, what is left out there? It is fibrous insoluble material. After biryani, you may like to have a tea, isn't it? But when you filter the tea, what is left out? That tea powder is mud, right? This is what is the mud. So simple clues will help you to remember those things. And next, menstruum, a liquid capable of penetrating and dissolving the drug substance. So it may be a water, it may be alcohol, it may be a glycerin. So which will able to penetrate and dissolve the drug substance is menstruum. Again, in making tea, the water or the milk act as a penetrating liquid media. Clear? Now coming to the mother tincture. Mother tincture pharmaceutically prepared according to old as well as new method by immersion. So what you are making tea is by immersion method only. If you keep the tea powder and the water for few days, tea will be made. It is like a immersion method. So that is what the mother tincture we do in old and new method. Mother solution, homogeneous mixture by dissolving. Simple salt solution, natramure. The crude drug name of natramure is salt, common salt. So if you make that solution is nothing but the mother solution. Next comes the mother substance, a solid mixture by trituration. So trituration, I will explain you that one. So it is for insoluble drug substances. So for that we go for trituration. In 2024, a crude mixture of mineral or organic material in thin pasty state, doughy mass, pasty mass is known as. Please answer. Please answer.
Magma.
Magma. Right. Very good. So the answer is magma. So this is what is the magma, and the supernatant fluid you can see that is what is the menstruum, and down you can see the mud. The answer is magma. Now coming to the old Hahnemannian method, classical method. So there are 9 classes are there. Class 1 to class 4 are mother tincture. Class 5 and 6 are mother solution. Class 7 to 9 are mother substance. So please keep it in mind, Hahnemann never wrote homeopathic pharmacy as a textbook. Whatever he has written, it is there in his source book like Materia Medica Pura, Chronic Diseases, Organon of Medicine, and few lesser writings. So in the Materia Medica Pura, in the Bellodana, he has written about class 1. In case of Tusa, in class 2, Scilla in class 3, Staphylococcus aureus and Spigelia in class 4. So these have been explained in Materia Medica Pura. Depending upon the nature of the drug substance, drug residue vehicle has been taken. So most juicy, medium juicy, least juicy, and dry. Again, you have to bahar the things. These all need to bahar. And coming to drug strength. So 1/2, 1/2, 1/6, 1/10. But you should understand what it is. So 1/10 means in 10 ml of the prepared mother tincture, 1 ml is the drug. That's why the drug strength of class 4 is 1/10. Okay. In class 3, 1/6. 6 ml of mother tincture is prepared. So in the 6 ml, the 1 ml is the drug. Like that, 1/2. Okay. So these all may be asked in the match the following cases, or they may ask you the source book it has been explained, or what may be the drug residue vehicle, what may be the drug strength. Clear? Right. Coming to the mother solution, class 5 and 6, aqueous and alcoholic by dissolution. I took the example of natramure, so where it requires least quantity of water, isn't it? Small quantity of water. But Hahnemann made mandatory to prepare by decimal scale. That is why 1/10. So 5A and 6A are in decimal scale, right? And 5B and 6B in centesimal scale. That's why drug residue vehicle is 1/9, and in case of decimal scale, and drug residue vehicle is 1/99 in centesimal scale. Okay, so again, natramure, acid phos, vacuum, mercur has been explained about these classes in Materia Medica Pura. Last, the mother substance, class 7 to 9 by trituration. So here the trituration method is used. The drug substances which are not soluble at all. Okay, so class 7, the minerals, for example, gold, which is not soluble. How many days you keep it in a water or alcohol or whatever, it is not going to dissolve. all the minerals, no source have been prepared in class 7. So this is dry, solid, insoluble drug substance. Please make it this one. The concept of the class 7. So fundamental rule again arsenic alba. It has been again explained in Materia Medica Pura. This can be prepared both in a decimal scale and centesimal scale. Drug raised to vehicle depending upon the decimal or centesimal 1:9 or 1:99. Now coming to the class 8. Here in the class 8, it is liquid, petrol, venoms, toxins, which are liquid in state, but still they are insoluble in a liquid media or liquid vehicle. That's why they have grouped under class 8, and the classical example or fundamental rule is petroleum, which has been explained in chronic disease. Clear? So again decimal scale or centesimal scale. Then last class 9. Here Hahnemann took all the fresh vegetables, fresh animals, whole living animals or dried animal living, all fresh one he took, part of the animal, lateral group, venue. So these were fresh one, and he took 2:9. But the drug strength did not change. Why? Hahnemann said that, so if you take two parts also, one part means half of the part will be lost while the preparation itself. So one part of the drug remains as it is. So he has explained agaricus in chronic diseases. So class 1 to class 9, you must remember all the fundamental rules and the drug raised to vehicle, nature of the drug substances, and which source books they have been explained by Hahnemann.
Ma'am, can you please repeat class 9?
Shall I do it at the end or shall I go now only?
Now.
Okay. Yeah. Okay, no problem. Class 9. Class 9 is the drug substance which are fresh in nature, which are fresh in state, like plant kingdom, whole living animals. Okay. So where spiders and insects, they are fresh. Okay. So those kind of drug substances he took in 2:9. Why 2:9 or 2:99? Because half of the drug substance will be lost while preparing, means while trituration. Trituration, you know, no? 60 minutes process. It has been divided into 20, 20, 20, where we are. Yeah. So in that trituration, half of the loss will be there. That's why he took 2:9 or 2:99 and explained agaricus
Fine.
in chronic disease. Any questions, please?
No.
Understood?
Understood.
Oh, clear. So in 2025, the Badiyaga mother substance is prepared under class 7. Please read the question carefully. The Badiyaga drug substance is dry, insoluble in nature in the light of above statement. Now, it's our human memory or human brain. We cannot remember all the drugs. We cannot memorize 5,000 drugs, which class or whatever it is. But if you understand the concept clearly, you can answer it. What was there in the class 7? What was there in the class 7? Please answer. Unmute yourself and answer it. It was what has been explained. It is dry, insoluble in nature. Class 7 is dry, insoluble in nature. What they have given for assertion? It is prepared. It is may be prepared, may not be prepared, shall prepare, something it is not there. They are very sure and correct sentence. What they have explained is, it is prepared under class 7. Clear cut. And their explanation, dry, insoluble. If you remember this one, what will be the answer? Which one is which option is correct here? Option number 3. Why? Both A and R are correct, and reasoning is correct explanation of the A. So, if you understand the concept wise, you are able to solve it, because we cannot remember. Badiyaga has not been explained in any of the classes here, but it has been included in class 7. How we can remember? Only by remembering the concept. I hope this statement will help you to understand. Right? Next, coming to the new. So, whatever class 1 to class 9 are, old method, and new method is which was being introduced by homeopathic pharmacopoeia of United States. There are two methods are there. Percolation, which is a short process within a 24 hours, means within a day we are going to extract the mother tincture, and maceration, which is a long process, so 2 to 4 weeks. The peculiarity of the new method is dry drug substance. In any condition, you must take the dry drug substance and uniform the drug strength. Uniform drug strength is there. How it was there in the old method? 1/2, 1/2, 1/6, 1/10, 1/100. It goes on varying with the classes. But here, what HPUS has written the uniform drug strength. Some variations are there. I will show you that variations. And if you remember that variations, it's well and good. And otherwise, all other drug strength is 1/10. Right? So, new method, maceration. I usually tell my students that this is an idli dabba, definitely. So, this is idli dabba only. Nothing more special it is there. So, what we are doing here? Hard, gummy, miscellaneous drug substances. Very hard, very gummy, which cannot be dissolved within a day or 10 days like that. So, what we are doing? We are extracting the active principles or constituents of the drug, remaining the drug in the room temperature with the solvent or with the menstruant, and we are frequently agitating it for 2 to 4 weeks, means for a month. So it is a long process. You should remember maceration as a long process, hard gummy miscellaneous drug substances, right? Requiring the time 2 to 4 weeks. Right. Next method. There are two methods only. Maceration, and next is a percolation. Soft, non-gummy, non-miscellaneous. For example, you take aloe vera in your home or garden, it might be there, right? It is very soft, right? Extracting that soluble constituents of the drug and preparing the mother tincture. But it is done with a special instrument called as percolator. So the percolator has got, so there is one toe is there. So what is toe means? What you have, see the arrangement of filter bed in your filtration of water. Remember, the thickness of the sands have been layered. You poured dirty water and you took clean, clear water. That is nothing but the toe here. The sand is also used. Green glass powders are also can be used. What we are doing here? So this neck will be locked and the percolator, the filter bed is arranged, and then the pulp of the drug is being arranged on over that, and menstruum, that is what? The liquid vehicle which has been added and kept locked for 24 hours and allowed to extract by in the receiver. So this is what? Percolation. Within 24 hours, we are going to get the mother tincture, whereas in maceration, 1 month. Remember that one. So again, questions have been asked in that. So the method used for preparing the mother tincture from miscellaneous gummy substance from vegetable and animal kingdom. Please answer this question.
Maceration.
Yes, it is maceration. Right, very good. Now, here, what is distillation? You must have heard the distillation of water, distillation of alcohol, getting the purest form of water. What is sublimation? Sublimation is what? Converting the solid into vapor state or gaseous state. Okay. For example, camphor is mixed with the soil and all. If you heat it, vapors of camphor will be pure form. And crystallization, crystals of pure drug substances. These all three are the laboratory methods to get the purest form of drug substances. You have done these practicals in your first year. So this is the answer is maceration. Next, in 2025, again, applied part of question paper. Applied part of the mother tincture preparation. Now, understand the question here. What they have answered, asked mother tincture preparation in new method. Means both. They are combined maceration and percolation, which is correct according to new method. Decantation prior to filtration is necessary in maceration. I have shown you idli dabba. Remember, you cannot extract like that. Okay. But in percolator, we have a receiver. But here what happened? There is no. We have to slowly decant it. So that is correct. And whether the percolation is a long process or short process?
Short plus 24 hours.
It's a short process. Very good one. So this is what? It's a short process. So that's why B answer is not correct. Powdered green glass in place of sand porous material. As I'm explaining percolation or percolator, the bed, both can be used. Powdered green glass as well as sand porous material. So this is correct, na? Right. This is correct. Gummy mucilaginous drug substances are percolated. You answer this question. Anybody?
Wrong.
Wrong. Why? Non-gummy, non-mucilaginous drug substances are percolated. Very good one. Next. Viscosity and adhesion are two physical forces. Now, when you have placed in the percolator above the filter bed or the sand porous, whatever the material toe was there, above that you have kept the drug material or magma. Above that menstruum is there. Which are the forces will be acting? First thing is gravitational force. Because of the gravitational force, the menstruum tries to come down to the receiver. Okay, but the viscosity and adhesion forces, physical forces of the perc will try to keep the menstruum. So this is also correct. So the correct option is A, C, and E. The second one. Understood, no? The concept. If you understand the concept, pharmacy, 60 marks in your pocket. Remember that. Give 2 to 3 hours. Next, in 2025, as I told in a new method, what is that? What is the uniformity here? Uniformity is 1 by 10. Right? So, here they have asked, it does not have the drug power of 1 by 100. What we have a knowledge? New method means always 1 by 10. So, some variations are there, I said, no? So, here one of the drug is there, which is 1 by 10, and all others, all three are 1 by 100. I would like to suggest you that if you remember, if you know that answer, then only attempt. Otherwise, unnecessary minus or negative 1 mark should not be there. So, that's why the correct answer is Phygelia, which has got 1 by 10. Okay, this has got 1 by 10. All other acid piclicum, gloninum, arsenic album has got 1 by 100. Even though it is new method, it is being prepared, but it is 1 by 100. Clear? Is it clear? Some other variations also we can answer. Now, drug power of the Moschus. We don't remember. What is the drugs? New method preparation. Again, drug strength they have asked. Again, when you go for it, what is new method drug strength? Usually we say 1 by 10. But it is wrong. If you do not remember Moschus, for which, what is the drug strength? Don't attempt it. Do not lose -1. So, correct answer is here 1 by 20. Perpetual sac of the musk deer has been taken and prepared the drug called as Moschus, where drug strength is 1 in 10 parts, 20 parts. That's why you have to remember new method. Do not go attempt directly 1 by 10. Think about it. Why they have asked Moschus. That's why if you know clearly and firmly, confidently, then only you go for it. Other variations like phosphorus, these all are new method only. Phosphorus 1 by 667, cactus grandiflorus 1 by 20 again, sulfur 1 by 5000. Please remember this type of peculiarities they will be picking, and these all are the phosphorus, cactus, sulfur, Moschus, and previous all three arsenicum, acid, then glonine, all those are also 1 by 100. Now coming to the sources of homeopathic drugs. Very vast subject. I cannot finish off in 1 hour. Not at all possible. Sorry for that. But if you remember few peculiarities of sources, definitely you are going to good score. So plant, already you know that one. Plant, animal, mineral, chemicals, no source are called imponderabilia, synthetic and allergens. Right? So one by one, I have tried to explain here aconite, belladonna, whole plant including root. Like there, I have given only 2. Many examples are there. You need to buy it. I usually tell my first year students to make a chart of it and make it on the wall. Make it as a wall poster so that when they are going in or going out, they will remember that one. If you have made like that, or you can make the flashcards also, then you can remember this one. Osmium sanctum, lobelia inflata, whole plant excluding root, all these examples. Here one point I would like to say that the part used in especially in a plant kingdom, that part is used which has what? Active drug constituents. Let us go back to our kitchen only. Which seeds are using, the coffee seeds are used to make the coffee, isn't it? We are not using the leaves over there because constituents are more active in seeds. We are using tea leaves, tea, chai patta. We are not using the seeds over there. Why? Constituents are more active in tea or chai patta. That's why you remember that. You remember that and study the peculiarity. Then you may get the good marks over here. Next, coming to the 2017, see here. We cannot remember all the drugs, as I told. At least if you remember, like coffea cruda, what part is used? Please answer.
Seed.
Seeds, right? Seeds. Asafoetida. Hing, what we are using? That is what? Is a gum resin. Only we know two. But other two we don't know or then do not accept. Okay. If you remember any of that one, Phytolecca decandra is what? Is a root. Fresh root is being used. If you remember three, fourth option will be again the whole plant. So what is the answer for this one? Answer 4. Etusa sinapum, whole plant. Asafoetida, gum resin, Phytolecca, root, coffea cruda, seeds. Right? So in this, especially in the sources of the drugs, if you are confident, please go ahead. Right? Then coming to animal kingdom. Again, animal kingdom also, many examples here, the whole living animals, dried animals, skeletons, the parts of the animals, secretions of the animals, venules, Ophidia group, and lac group is also used. So I have taken few examples, but you have to memorize all those drugs. But here one more thing, in the minerals, we know that aurum metallicum, gold, argentum, then all those platina and all. But here some mineral spring waters are there, which few are there you can buy. Saniquila, Levico, Vichy, then Pukamchup, then Weissbidan. Why these are important? You must have heard in Himalayan group that hot water, spring water is there, which is good for asthma and all. Like that, these all are there in Ottawa, South Tyrol, France, then Persia, and Pukamchup Lake, like that. Right? So, if you no need to remember about the location for your reference I have added, but remember the mineral spring water. They may combine or they may make a permutation and combination and may ask you which one is the mineral spring water or which is not the mineral spring water. They may go like that. So only few are there. Few, 3, 4 are I have left. You go through it in the mandal and mandal, you will get it. Coming to no source. No source is what? It is a drug which is prepared from the deceased person or from the deceased part or deceased product. I am directly going to the HPI classification, which has been explained in a 4th volume. How many volumes of HPI has been published? Please answer.
1984.
Very good. Okay. It was in 1971 in the first one. In 2025, online was available. PDF, it is there in the PDF, not at published. Okay. So, again, starting from N1, no source from N1. N1 means you remember endotoxin. Remember that the bacteria which produces endotoxins are included in N1. So example is typhoidinum. N2. Just remember microorganism capable of producing exotoxins. The example is diphtherinum. Remember this. And N3 is purified toxins. Means it may be N1, it may be N2, the bacteria, but these have been purified and cultured and then they are prepared into a medicine. So tetanotoxinum. So the shell life of N1 to N3 is 6 months. Okay. But N4, which has been prepared from the clinical material from the deceased subject, means from the patient only. For example, sorinum has been taken from the scrapings of psoriatic skin. Like that it has been prepared, right? So tuberculinum is from the sputum of the tuberculosis. So like that it has been prepared. And the shell life for this one is 12 months are there. For N1 to N3, 6 months is there. So coming to the questions, microorganisms, viruses, clinical material from human subjects. Which one? What is the option? Quickly answer. We need to solve more questions for you. So this is confusion question because microorganism is there. So you may confusion with the N1, N2, N3. But think about clinical material, N4. Clinical material is N4. So no sort 4. Right? Like that you have to understand the question properly. 1 minute is given. 60 minutes in that 120 questions, no? So 60 minutes, one question for 1 minute. So you have to understand and you have to react back. And then coming to the sarcodes. Sarcodes means healthy animal tissue. Right? So many examples like orchitinum, mucerinum, peltor, all these may you get in the animal kingdom also. Right? All sarcodes belongs to animal kingdom. But all animal kingdoms are not sarcodes. Why? Because sarcode is a healthy tissue. Healthy animal tissue. If it is healthy only, it is being prepared into a sarcode. So disease or something affected part, the secretion are not taken here. So here only healthy animal tissue has been taken and prepared into sarcodes. Next, coming to the vehicle chapter. Again, vehicle, material, agent, therapeutically inert, chemically neutral, non-reactive with the drug substance, harmless, edible, palatable, preservative. Types of vehicles are solid, liquid, and semi-solid. So big chapter is that. Big means not big as anatomy, physiology, pathology like that. Few chapters, 10 to 12 pages are there. You can memorize it. Don't worry. So here which I have marked with the red, no? Sugar of milk, purified water, alcohol, spermicity, lanolin. This will be asked in the properties. They have been asked in the properties. Okay. So please go through this one, these properties, memorize it because from 2017 to 2025, only these have been asked. They may ask any other question also, glycerin also, cones also, tablets also, but these have been asked. More priority first study this one. If time permits, study all other things. Okay. Next, going to the sugar of milk. Again, sugar of milk is source is goat's milk. Formula C12H22O11 water. Then milky white, odorless, faintly sweet and sandy touch. Molecular weight. Solubility property has been asked in the questions. So 1 gram of sack is soluble in 5 ml in 2.6 ml of boiling water and it is totally insoluble in alcohol. So the question has been asked in 2018. What is not? Negative type of question. Please remember this is what? Not. So negative type of question. Sugar of milk. So answer please. What is the answer? Okay. 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 So it is not soluble, insoluble in alcohol. Remember that. Okay. Next, coming to ethyl alcohol or ethanol, grain alcohol. So this has been prepared with molasses, rich in starch and sugar. Synthetically you can prepare. Remember the formula, molecular weight. Alcohol is a good solvent and preservative. That's why our all preparations are from the alcohol. Unless and until we say that when which type of alcohol, otherwise it is being prepared in ethyl alcohol, that too in a strong alcohol. Right. There are varieties of alcohol are there. In that, strong alcohol is used to prepare the mother tincture. Right. So dilute of alcohol for the first potency, dispensing alcohol for the higher potency. After the preparation of first potency, second, third, fourth, like that, they have been used for the higher potencies. This you have to remember. Next comes one super calculation. This you have to remember. It has been asked. In proof spirit means the 57.1% of ethyl alcohol and rest is the purified water. The alcohol which is stronger than 57.1 ethyl alcohol is over, and which is less than that is a under proof. Right? So there is a small simple formula is there. Conversion of percentage to proof spirit. They may ask you, what is the proof spirit of 90% of the alcohol? So the formula is 90 into 1.75 minus 100, it equals to 57.5. Right? The answer is in a positive integer. The answer is in a positive integer. Remember that. Then it is overproof. Next comes the 50%. Here 50 into 1.75 minus 100. The answer is in a negative integer. That's why it is underproof. Right? So percentage into constant 1.75 minus 100 will give you the percentage to proof spirit. It will help you to calculate. Next, they may ask you proof spirit to the percentage. They may give you 60° OP, overproof. Then if overproof is mentioned, you have to take this 60 as plus 60. Plus 60 plus 100 divided by 1.75. Answer is 91.45%. If 60 UP has mentioned, both it is here OP, here it is UP. It is negative. Once it is UP means it should be negative. So 22.86%. Plus sign you have to use with the proof. If overproof, if it is mentioned UP, you have to minus it. The formula for plus OP and minus for UP plus 100 divided by 1.75. Now, I will ask you to calculate the 80% of the alcohol concentration. Remember the formula? Are you active? What was the formula? Percentage into 1.75 minus 100. Don't worry, all overproof have been given. Any one might be right. So, correct option is 40° OP. Third option. Okay. So, if you simple formula, one formula is there in homeopathic pharmacy. So, remember it. Then prescription. Prescription here, already we know, written document, give a physician to the dispenser and preparation of the medication and instruction to the particular patient. RX for sign of the Jupiter where recipient receive has been mentioned. Then there are 4 parts: superscription, inscription, subscription, signature. Superscription is preliminary data. Remember like that. Preliminary, patient A, sex, address, instead of that one, if you remember, you already are doctors. So you know that one, preliminary data. Inscription means the remedy and the vehicle. Remedy and the vehicle. Potency, quantity, everything. Right? So inscription. Subscription. It is for the dispenser. What he has to prepare. That is what is the subscription. Coming to the signature, where mode and time of intake of the medicine, when to report, advice, signature of the physician and registration, which is with the for the direction of the patient. Only signature part is for the patient. Subscription, dispenser. In 2020, ideal prescription consists of 4 parts: superscription, inscription, subscription, signature. Correct? Yes or no? Please unmute yourself and answer. Right? Then signature part. Directions are given in the which part? Signature part. So the option is both statement 1 and statement 2 are true. Option 2 is the right answer. Coming to potentization. Coming to here, it is a mathematico-mechanical process by which latent curative properties of the drugs are earned. So common salt, natramure. Common salt, just nausea and vomiting. If you take the salt solution, in natramure 1 ml cures migraine. So that is what curative properties of the drugs are. So in Organon of Medicine, in 5th edition, it has been explained. 2 process, trituration, insoluble substance, succussion, soluble. So trituration is up to done 3C in case of centesimal scale, 6X in case of decimal scale. Thereafter, they have been prepared by the method of succussion. If you remember that much, enough here. Then, who discovered centesimal scale? Dr. Samuel Hahnemann. And decimal scale, Dr. Harry. 50 millisimal scale, Dr. Samuel Hahnemann. But the name has been given by Dr. Perez Smith. Okay, drug ratio 1:99. Already you know 1:9. Here in the 50 millisimal scale, 1:9. 50,000. But here, while doing the trituration, the division of lactose has been asked. So according to Hahnemann, it is always equal. So these 99 parts are being divided to 33, 33, 33 or 9, 3, 3 raised to 3 raised to 3 raised to. But here in the HPI, 11 for 33, 55, 1 raised to 3 raised to 5. So 2018, who has introduced? If you remember, organelle person, Dr. Constantine Herring, who discovered decimal string. Right? In 2025, the sugar of milk, HPI has divided. So all HPI means option number 4, 1 raised to 3 raised to 5. Coming to legislation, this is little complicated subject, but you have to memorize it. So what has been written in a red mark now, that will be asked in the MCQs. Most of them they have asked. So April 10, Honey's birthday, Hahnemann's birthday, 1940, and drugs and cosmetic rules, 1945, and all part 6, 7, and 9 deals with homeopathic medicine, and schedule M1, which is about the GMP, that is factory premises for the manufacture of homeopathic preparations. And these all are part 4, part 6, and the rules for the import and the registration 30 and 32A has been used, and for sale of homeopathic medicine part 6A from 67. If you remember 67, they may give the, for sale of homeopathic medicine, they may give the 30, 32, 67, 80, they may 85, they may give the option. So the answer is from 67A to 67H. Then coming to the part 7, manufacture of sale of the homeopathic medicine. Do not confuse with the sale of the homeopathic medicine there and here. Here is the manufacture part. So 85A to 85I. And labeling and packing, 106 and 106B has been asked how we have to label it. And standards for external application, especially ophthalmic preparation, external application, has been explained in 126A. Clear? So some in-depth questions they have asked, that is, what are the rules for the quantity and percentage of ethyl alcohol? That has been explained in 106B. Right? So these are the reference book. What I suggest is simple books, recommended focus area. If you focus it within a day or 2 days, you can complete that one, and you can refer back in the YouTube and come back and study in that manner. Definitely it is going to help you. Other than this, what all are the MCQ reference books are there, that also you can study it. So I would like to thank our management, our beloved principal, Dr. Anand Vasur, and our beloved madam, Dr. Vijayalakshmi, PG coordinator, and all other my staff and colleagues who helped me to prepare this PPT as well as to present this one, and who are there with me now at this time also as a moral support. I thank Dr. Halikeri sir, Shushmita, and our PG student, Dr. Kavya. And so thank you and all. Just some few questions which are out of the box will be explained by Dr. Shushmita. She's a good candidate as she has finished the MD Pharmacy. You may ask and you can clear the questions with her. And if you have any questions, please. Otherwise, I will hand over to Dr. Shushmita. Okay? So she will be solving previous year question papers. Shushmita, please. Good evening, everyone. Am I audible? Am I audible?
Yes, madam, audible.
Yes, ma'am, you're audible.
Thank you. Okay. Till now, we have discussed in detail about the homeopathic pharmacy topics. What are very much important, very much weightage topics for your AIAPGET examination. And now we'll discuss about few of the questions that are, I have collected these questions purely from the question papers from the year 2020, 2017. And 2025. Okay, we'll start. Be active in the group. You can use the chat box for your comments. If you have any doubts, you can ask. Okay, the first question. All of the following are prepared by the process of trituration, except they have asked negative kind of question, that is except. Go through the options. You can answer in chat box. Okay. Some are answering option 2. We'll see the correct option. By seeing this picture, can you answer now? Yeah, the answer is cinchona. Because everything is prepared according to trituration, cinchona is prepared by the process of mother tincture preparation, not by trituration. Okay. Again, the 2017 question. They have asked about something like match the following. Okay. In the list 1, they have given the drug names, and list 2, they have given the alkaloids present in the drug. You have to match the list 1 to list 2. Okay. The options are like this. Belladonna, everyone knows which is the alkaloid present, and Epicat, everyone knows. These are the common. Okay. Answer, you can unmute and answer also. Answer is 2. That is, everyone know, I think. We'll move to the next one. Okay. The question is, this is same. The first question and this question also matches. Just go through the question. Which part of the plant Cinchona officinalis is used? Everyone knows from the first year onwards the cinchona bark experiment. In that only it is there. Answer is option 1, bark. Okay. We'll move next. The question is from 2023. Here they were asked like arrange the number of monographs according to their chronological order of publication of Homeopathic Pharmacopoeia of India. This is like we have to arrange according to their publication years. Can anyone make a try? This is little tricky question. You have to know all the publication years. Can anyone make a try? Okay, we'll go to the answer. The answer is answer 2. It is like first one is from the E, that is 100. It came in the year 1974. And B, that is 1984. Next one is like 1986, that is 114. Okay, clear? Don't be confused. The 100 monographs are there in 2 years. Like first one is in the year 1974 and one more is in the 2000 year also. Okay. Next question is, it is about potencies. That is, according to Hahnemann, which scale of potency is capable of producing the medicine of highest development of power and mildest action? First is like stride potency, jumping potency, decimal scale potency, and 50 millisecond. If you use a common sense, you can answer this question because Hahnemann has introduced only 2 scales of potencies. What are those? First one is decimal scale, and next one is 50 millisecond. By this, you can answer, that is 50 millisecond. Okay, clear? What is stride potency? Stride potency is nothing but conversion from 6x to 7x directly. That is with the water dilution. Jumping potency is from 6x to 8x. Clear? Decimal scale is introduced by Dr. C. Herring. Okay. Okay. By this picture, can you identify anything? You can unmute and tell. Okay, we'll go. Who authored the first homeopathic pharmacopoeia? When the in the year 1825, you can make a try by this. Can anyone?
Yes,
Someone
ma'am.
answering? Okay. The answer is A. That is Dr. Carl W. Caspary in the year 1825. Clear? That is Homeopathica Dispensatorum. Book name. Next question. Here they have asked about the cones. Cones are made from what? They give the options: sugar, milk sugar, mixture of globule and distilled water, mixture of cane sugar and egg albumin, and dextrose. Can anyone? Okay, some are answering C. We'll see the answer. The correct answer is mixture of cane sugar and egg albumin. This is only one vehicle which uses the two kingdom, like from the egg albumin and from the cane sugar also they are preparing. Cones are used only for keeping the drug for a longer time. Okay. Here again they have give the match the following, like list 1 and list 2. In the list 1 they have give the drug's name, and in the list 2 they have given the classes which belongs. This is typically of the following Hahnemannian old method in the fundamental rules. These drugs all are there in the fundamental rule according to their classes. The options are like this. Can anyone make a try? You can unmute and tell also.
3.
3.
3. Okay. We'll see. Option 3. Correct. Very good. Okay. Next one. This is the potency they have asked. In 1 lakh potency, centesimal scale is designated as what? Okay, some are answering C. Okay, the answer is C, that is cm. Cm is termed as 1 lakh potency. Next, again, they have asked about the match the following. It is like in list 1, they have gave the classes, and list 2, they have gave the drug's name. In this, the class 5B is about mother solution. The remaining are class 4 is about mother tincture, class 9 and class 7 is about mother substances. Okay, here only fluoric acid that belongs to mother solution. The first one is there. Then the options are like this. The correct answer is answer number 2. Okay. Next one, the number of monographs in homeopathic pharmacopoeia of India in the volume number 4. Volume number 4, when it came? Okay. Some are answering correct. Number 3 is the answer. 107 monographs are there in volume number 4. Next question. This is little tricky question. Like what is the life period of nosodes below 3x of group N1, N2 and N3 from the date of its manufacture? They have asked only about N1, N2 and N3. Okay. Anyone answer? Okay, the correct answer is 6 months. It is mentioned in the HPI volume number 4. They didn't mention anything about N4, but only N1, N2 and N3 has the life period of 6 months only. Okay. The picture, can you identify any drug? Okay. The part used in the preparation of mother tincture of Azadirachta indica. Which part is used? Can anyone make it right? Please answer. Some are answering option 3, leaves. The picture indicates more of leaves, that's why it is leaves. Okay. The correct answer is bark. Bark is used in this. The next question, it is little about standardization. The following parameters are used to assess the quality of the mother tinctures, except they have given 4 parameters. In that 3 are used for to assess the quality of mother tincture, but 1 is not there. Which one we have to identify? What are total solids? What is UV absorbance? What is weight per milliliter? What is water content? Can anyone answer this question? Water content is the answer, because in the assessment of quality of the mother tincture, we use total solids, UV absorbance by the fluorescence analysis and all, weight per milliliter by the use of specific gravity bottle, but water content we are not going to see because we are using in the strong alcohol, that's why. The next question we'll see. Okay, this is about the Drugs and Cosmetic Acts. They have asked like which schedule of the Drug and Cosmetic Act, Rule 19, 45 provide the rules regarding the GMP, that is good manufacturing practices, and requirements of the premises, plants and equipments for the homeopathic medicine. They have given schedules. If you know, if you go through the DNC Act, it is very easy. Only one schedule is there relating to the homeopathy. The answer is Schedule M1. Only Schedule M1 is related about the homeopathy. Other schedules are not related to homeopathy. Okay. Again, they have asked about active principle or alkaloid. The following active principles are found in Atropa belladonna, except they have given 4 active principles. All the 3 are belongs to Atropa belladonna, but only one is not belongs to Atropa belladonna. Can anyone? Yeah, some are answering 2. Okay, we'll see the correct option. The correct answer is 2, that is emitting. Where emitting is present? Can anyone unmute and tell? Where emitting is present? Before we have gone through this emitting in the match the following question. Second question was on this only. Emitting is one of the main alkaloids found in the EPICAT. EPICAT roots we are using in that EPICAT. Emitting is the main alkaloid. Okay. Next one is about pharmacopoeia again. Arrange the following pharmacopoeia according to their chronological order of the publication when they have published. These both official and unofficial both are there. Okay. The option is like this. Can anyone make it right? If you know the year of publication of each pharmacopoeia, you can answer this question. Okay, we'll see the correct option. Some are answering 1, 1. We'll see. Answer is 2. Because they have asked like the BHP, that is British Homeopathic Pharmacopoeia, came in the year 1870. Next comes the GHP, that is compiled by Wilmer Schwabe. First GHP came in the year 1825, agree, but compiled by Wilmer Schwabe in the year 1872. Next comes the American, that is in 1897, and by the M. Bhattacharya, that is in the year 1892. Okay. Next question is about the sampling methods. Where the sampling methods, while reading for the first year, we skip this sampling, not much important and all, but here they have asked this 1 year. When the total weight of the drug is to be sampled, that is less than 10 kg, the final official sampling weight is less than what? Less number of kgs are there, so it may be less. So go through this. Okay, the correct answer is 125 grams. Okay. When the weight of the total drugs is more than 100 kg, it should be 250 grams. When it is less than 10 kg, it should be the 125 grams. Clear? Okay, we'll move to the next. By this picture, what you'll identify? Can anyone unmute and answer? What is this picture? Any vehicle? Okay, this is lanolin. They have given the properties of the lanolin, melting point, the water content, and also the solubility they have given. You have to choose the correct options in this. The options are like this. If you know the properties of lanolin, you can answer this. Okay, some are answering 2. Okay, we'll see the correct answer. There, 2 is the correct answer. That means A, D, and E is only right according to the lanolin. The melting point is 36 to 42 degrees Celsius, and it contains not more than 0.25% of the water, and it is freely soluble in solvent, ether, and chloroform. The source of lanolin, can anyone answer? It is from the wool of the sheep. Okay. Okay, we'll move to the next. Okay, they have again asked about the preparation of drugs. The following homeopathic drugs are prepared under class 5 method, but except in this they have given some 4 drugs. All are prepared in class 5A, but one is not prepared under class 5A. Which is that? Can anyone answer? This is very easy. If you know about acids. Okay, the correct answer is acid benzoicum. That is benzoic acid. That comes under 6A. Okay. The next question is about the imponderabilia. What are imponderabilia? It is one of the main source. They have used non-viable source it is. Okay. Which of the following is an artificial source of imponderabilia? Here they have given 4 options: magnetis polus australis, magnetis polus ambo, and solo, and radium bromide. Okay. The answer, some are answering it is option 4 and all. We'll see the correct answer. Okay, the correct answer is radium bromide. Radium bromide is an artificial source of imponderabilia. Other 3 all are the natural source of imponderabilia. Okay, good. We'll see. This is very easy question. The mother tinctures are prepared by using which type of alcohol? Very easy. Can anyone answer?
Strong.
Okay, strong alcohol they are answering. We'll see the correct answer. That is strong alcohol. Okay, this is about the terminologies. Which of the following branch of pharmacy deals with the repetition of doses? The options are there. Okay, they are answering A. Okay, we'll see the correct answer. The correct answer is A, that is pharmacophilaxis. That means the repetition of dosage. Then pharmaconomy is about the routes of administration. Okay. We'll see next question. This is little tricky question. This is like for the preparation of mother solution in 50 millismol scale of potency, the ratio of alcohol to water and the strength of alcohol. They have asked about mother solution for the 50 millismol. For 50 millismol, we start from 3C. From 3C, we prepare mother solution. Then we go to 0/1, 0/2. They have given the ratio also and the percentage of alcohol also. Can anyone make it right? Some are answering B, 1. Okay, we will see the correct answer. That is 1:4 ratio and 95% of volume by volume. 1:4 ratio is one ratio of the alcohol and 4 ratio of distilled water has to be used. Okay. The next question is about drug power or drug strength of class 3. Very easy, direct question from the old method. Class 3 drug strength. What is the drug strength of class 3? B. Okay. The correct answer is 1/6. B. That is the correct answer. Okay. We'll go to the next question. They have asked about cones. They have asked about the 50 millismol scales. The first, they have given some of the statements and which is related to 50 millismol scale, we have to see. First one is cones are used as a vehicle. Is we using cones in 50 millismol scale? No, we are using poppy-sized globules. So this is not correct. So the correct answer is B. That is B, C and D are correct. That is Dr. Hanuman has termed the new preparation as Medica a globule. That means we are using globule here. So he has termed as Medicamenta a globule. And C is the name 50 millismol scale was coined by Perry Smith. That is correct. And the D, that is as per HPI instruction to start the process of preparation. The scale was introduced in the 6th edition of Organon. Till 5th edition, it was not introduced. And in the 6th edition, they have introduced this scale. Okay. The next question is about the impurity test of the vehicles what we are using. This is according to that. First statement is oil of wintergreen test is used to detect the presence of methyl alcohol in the ethyl alcohol vehicle. The second statement is about iodine test is used to detect the starch in the sugar of milk. Is both the statements correct or wrong? Both the statements are correct. Okay, both the statements are correct according to their impurity test. The correct answer is answer number 1. Both the statements are correct. Okay. The next question is about who proposed the double-blind method of proving in drugs in the healthy volunteers. It is about the drug proving, where in the new HPT, that is homeopathic pathological trials, what we say, or human pathogenic trials, in that they have introduced the double-blind method. That is neither a master or nor the prover knows about the drug, who is taking the drug. That is called a double-blind method. Who introduced this double-blind method? Is Herring or von Heller or Treslev? Who introduced this? The correct answer, some are answering 3. Okay, we'll see the correct answer is 3. Okay. The next question is about the legislation. Here they have given the certificate of renewal to manufacture for the sale or distribution of homeopathic medicine is issued in form. Which form? There are many forms in the Drug and Cosmetic Act for the receiving, for the application, for the renewal, and all are there. Here they have asked to renewal to manufacture for the sale, for the distribution of homeopathic drugs. Some are answering 3. We'll see what is the correct answer. The correct answer is form number 26. Okay, I'll tell little bit about the forms. Form 24 is for the application of license. You are applying for the license to get the manufacturer. Next one is form number 25. This form number 25 is you are getting granted license to manufacture the homeopathic medicines. And in form number 26C is about renewal of the granted application. Like you have already got the certificate to manufacture. The renewal after the duration of 5 years, we have to renew the certificate. That renewal has to be done in form number 26. Next one is form number 20C. This form number 20C is about license to sell the homeopathic medicine, not for the manufacture, to sell the homeopathic medicine. Each forms will have different points about the sale or to manufacture and all. The duration will be 5 years mostly. Okay. This completes the question and answer session about the homeopathic pharmacy. Is there any doubt? Any doubts about regarding this? Is there any doubt? If not, we'll conclude the session here. Thank you for attending the session. I thank my management, our principal sir, Dr. Anand Husur sir, and my HOD, Dr. Manjula ma'am, and for the PG coordinator, Dr. Vijayalakshmi ma'am, and also for the team Jignasa for giving this opportunity. I'll thank everyone and we'll conclude here.
Thank you, ma'am. Daneshwari, over to you.
On behalf of Jignasa Karnataka, I sincerely thank Dr. Chitra Sivai ma'am, Dr. Manjula S. Halehuli ma'am, and Dr. Sushmita KB ma'am for sharing their valuable knowledge and inspiring guidance during the AIAPGET exam orientation session. Your insights have motivated us and will be greatly helpful for us in our examination preparation. I also thank every participant for making this program meaningful with your enthusiastic presence. We look forward for welcoming you again for many more enriching sessions in the future. Thank you all.
Thank you, Jignasa team. Thank you.
Thank you, ma'am.